All right, hi everybody. This is me without my mask on. So I'm going to make some supplemental videos for your class to help you with some of the processes that PowerPoint just doesn't do justice for. One of those is early development of humans. So that whole process that we talked about from lecture 4.1 to 4.2. My little video floating screen that I have right here, I'm going to actually put that away as I start to draw stuff out so that I'm actually not in the way. What you want to do is maybe download this supplemental file that I've included as part of this video and follow along with me. Print it out, put it on your tablet, and actually draw out the process. Don't just watch me blindly and try to memorize it. It's very helpful if you can do it yourself. Get yourself a whiteboard, draw it out on the whiteboard, erase it, and draw it again. So we're going to start with what are the objectives for these videos. I'm going to try to cut my videos into maybe no more than 10 minutes long. We'll see how well that works because sometimes I talk a little bit more than I expect to be able to, or I expect to be talking. So the first video, I'm going to show you the process for about the first 3 weeks of embryonic development. And then the next section, we're going to look at trilaminar embryo complication, how to build your basic cross section, and we're going to go from there. That's why you want to like pause, grab the supplement you can right now and follow along with me. I recommend some colored colored pencils in order to do this as well. So we're going to start out with this 3 weeks, what's happening during the first 3 weeks of embryonic development. All right. So why is this important? Okay, so I talked about this in class. This whole idea is important because I want you to be able to take this cross section and figure out what everything is in this cross section. And you can't do it necessarily right now, but by the end of 2240, you should be able to see some of the things that is in this cross section and kind of have an understanding of where this cross section is taken in the adult form. So we're going to do this again at the end of these cut videos. So the first thing is for embryonic development, we're going to start at the very beginning. So here I have my circle, and my circle is going to represent my egg. Okay. Remember, the egg and the sperm have to come together during fertilization. And our egg and our sperm only have 23 chromosomes. Remember, egg comes from the mother, sperm comes from the father. When they come together at fertilization, hopefully you take a pause and you're like, what do I create next? I'm going to create the zygote. Okay. So the zygote, it has 46 chromosomes. Remember, half from your mom and half from your dad. The other thing is, as soon as that sperm enters into the egg, this black line that is the circle for my zygote is going to become my zona pellucida or pellucida. Remember, that's that hard structure that goes around the zygote and prevents the penetration of more sperm. So going from a zygote to our next phase, the next phase, hopefully you know, or the next structure, hopefully you know, that is the morula. So we are going to remember that the process to go from one cell to multiple cells is cleavage. That's what you'll write right there. Remember, cleavage is basically mitotic divisions or mitosis. And cleavage is going to be holoblastic. So what that means is, you know, we're going to have one cell, and that one cell is going to split completely into 2 cells. Those 2 cells are going to split completely into 4 cells until we have our morula, which is going to be a solid sphere inside of the zona pellucida, which has 32 cells, right? So remember, this equals 32 cells, and it's solid. The zona pellucida is still around it. This is why the zygote and the morula are the exact same size. Remember, you're not feeding at this time. The cells just have to get smaller and smaller and smaller in order to fit in that sphere that is the zona pellucida. So after that, the next stage, we're going to go to our next slide, and we're going to see that, okay, so over here was the morula, right? The morula is then going to go through further development, and essentially what's going to happen inside of the zona pellucida is that our morula is going to become a blastocyst. So the cells that make up that not hollow sphere, that solid sphere are going to start to migrate to the outside layer. So this little squiggly line, just think about each one of those little circle things is a cell. That outer layer becomes the trophoblast. Remember, this is our invisibility cloak to make sure that it embeds into the endometrial lining. And then we're also going to cluster the cells that are in the rest of our morula towards one side, one edge of our new structure. And remember, those are going to be the inner cell mass, which is also known as the embryoblast. Okay, when we do that, we create a fluid-filled area called the blastocyst. That is going to be the area where stuff is going to diffuse through, and basically it's going to also allow for the growth of our embryo. We need that area to grow in. If we're solid, we don't have a place where we can add more cells. This structure becomes the blastocyst. Do a little eraser right there. Okay. The blastocyst is going to go through implantation here. And once it starts to implant, you're going to start to create your bilaminar embryo next. So implantation means that my zona pellucida has to hatch away. So instead of having that black layer, I'm going to replace that black layer with my kind of reddish brown layer here, which is my trophoblast. Remember, it's the trophoblast layer that's going to insert into the endometrial wall, which is the wall of the uterus. So here I'm going to build my endometrial layer. And then the trophoblast actually thickens like that. It's going to thicken as we implant. So at this time, you're still going to have the inner cell mass here. Remember that inner cell mass has to be oriented or pointed towards the endometrial layer. And I'm just going to abbreviate. Don't ever abbreviate on a test, okay? I'm just going to do it to make sure that I am within my 10-minute time frame. We still have our blastocyst. So remember, this area is the blastocyst. Okay. The next phase is that we're going to start to create our bilaminar embryo. So I'm just going to write that right there. What's going to happen is I'm implanted all the way or almost all the way. And when I create my bilaminar embryo, again, I'm still going to have my trophoblast layer on the outside. Just like that. But now my inner cell mass has to differentiate into two specific layers. So we're going to do this really simplistically to begin with. We're going to create one layer that's blue. That layer is going to form my epiblast. So what happens is the inner cell mass, the cells start to kind of separate into two layers. One layer that's going to be on the dorsal side of our embryo is the epiblast. The other layer, which I'm going to make in a lighter green color because yellow is kind of weird here, is going to be like this, and that creates my hypoblast. The hypoblast is going to be on the ventral side of the embryo. And if you're wondering where the rest of the trophoblast is, the trophoblast is going to be all up here. And then, of course, the endometrial layer is around that. So you want to remember that the blastocyst is what's implanting into the endometrial layer. And as implantation happens, we also are creating the bilaminar embryo. So the next phase is to take our bilaminar embryo and make it more complicated. So we're just going to write bilaminar embryo again. And again, I'm going to draw over my black circle so I know my zona pellucida is gone. And at this point, you're probably completely embedded in the endometrial layer. I'm going to first build my layer of epiblast cells. There's no difference there. And I'm going to lay down my hypoblast layer. Okay, you're still going to have your coelom. If we go back here, we still have, not the coelom, excuse me, the blastocyst. That's space. What we're going to do now is create the amniotic cavity and the hypoblast. So this kind of is one of those things that gets you guys a little bit. On the dorsal side, our epiblast cells are going to start to move dorsal just inferior, or deep, I should use deep to the trophoblast, and they're going to come together and create the amniotic cavity. So remember, that's the fluid-filled cavity that's going to protect our growing embryo and protect our growing fetus. At the same time, our hypoblast cells are starting to move towards the ventral side of the bilaminar embryo, and they're going to create the yolk sac. So you see how the yolk sac goes into the blastocyst area right here? That's what that is for. So I have an area so I can grow my yolk sac. So let's draw this just really quick so you also understand that the bilaminar embryo is two flattened discs. So if we're looking at the dorsal side of our embryo, we're going to have a flattened disc with our epiblast cells here. And then underneath it is going to be our hypoblast cells. So there's my hypoblast cells. Remember, we're looking at this from the back side, from the dorsal side. So think about, I'm looking at it from that side. I cut off my amniotic cavity. And you can't see the yolk sac because it's on the other side of my screen. If we look at this in a sagittal section, the sagittal section is not going to look a whole lot different than the transverse section up here. But you kind of basically are going to create your amniotic cavity just a little bit bigger, and there's my epiblast, like that. My hypoblast, what I'm going to do is make my hypoblast a little bit lower. I can just go back and erase later. So my hypoblast is going to be just underneath my epiblast layer, and then my yolk sac kind of starts to form like that. So it looks the same in a sagittal section versus a transverse section. What's going to be different is we're going to use that sagittal section to help us with the next part. So the next part of my video, I'm going to go over how we go from a bilaminar embryo to a trilaminar embryo. And that's through the process called gastrulation. Okay, so we're going to pause right here. Let's see if maybe you can do this yourself and then come back to the video if you need help drawing out everything from bilaminar to trilaminar embryo.